Hello everyone, my name is SMorgan, and today I'm going to tell you about my internships where I studied virology and pathology. But first I'm going to tell you about why I chose virology. The two images on the screen are of my favorite two TV shows, Helix and The Last Ship. Both shows focus on a virus entering the human population, and in the case of Helix, it's a man-made virus named Narvik C, and Last Ship is a virus that's sort of similar to Ebola. And this got me thinking about why not pursue an internship in virology. And so eventually I ended up finding my mentor, Dr. Judith White, and she studies the cell entry methods of Ebola. And she is a professor of cellular biology at UVA and a professor emeritus at Harvard, which is her alma mater. Dr. White's lab is located in Charlottesville at Jordan Hall, and her lab is on the 4th floor. I'm not a fan of elevators, so I took the stairs and I got lost. I walked around for 30 minutes until finally an undergrad found me wandering around, and she showed me where the lab was. And on the outside of her lab, you can see all of Dr. White's work, and there's TVs that show you all the progress that they're making. And you can look at from the outside of the lab the work that they're doing on the inside with Ebola cells. So the first time that I walked in, it's like a science nerd's heaven. So for me, it was like heaven on earth. There's wall-to-wall centrifuges, pipettes, all these cell cultures, and there's this big area that's secluded from the rest of the lab, which is where they house the Ebola. They have synthetic Ebola and real Ebola, which is similar to what the CDC has, which allows them to do their research. And Dr. White's lab, her office is located near that area so that they can, if something happens, they can immediately shut things off and have the whole area locked down, and it's a huge, complicated process, which I learned about as a first aid. There's 4 classifications for Ebola that they use at the lab. If it's a class 2 and below day, I'm allowed to be in there, but if anything's above a class 2 virus that they create, I can't be even in the building. It's only hired personnel and trained staff and EMT responders in case something happens. So it was a class 2 day, and they were currently working on some of the research that they were doing with birth control medicine and fertility hormones to stop Ebola's receptors from entering the cell. Ebola looks like, under a microscope, Ebola looks like a worm. And I was able to look, Dr. White's undergrad was on the other side of the wall doing cell cultures. And it's this cool microscope where Liz has the cell cultures and the slides and all the different areas that they have to put Ebola in on her side. And then the other side, there's these little outlets that have like a sort of the end of the microscope where you can look in without being in there. So I was able to watch an Ebola virus enter the cell, and it's released its cytokine storm. And a cytokine storm is sort of like a thunderstorm, except instead of electrons flying around in the atmosphere, you have Ebola particles. And that those streak of lightning that you see is how Ebola enters into more cells and into the bloodstream. And the overall experience was amazing. I did a lot of reading because I can't really touch Ebola because it's Ebola. So I did a lot of research and reading and looking at data, which is how I came up with my research question. Does the US have the potential of being affected by a virus that causes a large-scale outbreak? And the answer to that question is yes. We're a social nation. We have millions of people in our country. We have different social patterns. We have different travel patterns. And we have over 40,000 ports of entry into the country. And we also have different socioeconomic statuses, which is what the factors that my research paper focused on. If you think about it, you're all sitting beside somebody who's breathing out different virus particles and bacteria. Sorry, guys, I'm freaking you guys out. So, like, as you breathe in and out, if a person was to say have Ebola, like Ebola Guinea wasn't airborne, but Ebola Reston, which was here in the 1990s, which we learned about in our hot zone, for anybody who got through school. If that virus was to jump over to the humans, it would be airborne. And so somebody someplace breathing can infect a whole room of people like here today. So imagine a city like New York City, huge, and you have LaGuardia Airport and all these major points of entry and different points for sightseeing. And you have millions of people traveling in and out of the country through that one location, and all those people have the potential of bringing in a virus into the country. And if, say, for instance, with Ebola, if an Ebola person from New Guinea would come over to the United States with a simple prick of their finger on a cut cab seed, they can spread the virus to anyone who comes into contact with that cab seed, and anyone who they come into contact with would spread the virus. So it's just a continuous trend of people who potentially can become patients or potentially can become vectors for spreading the virus. So it's a big world that we have out here, which is very scary when it comes to virology. Unfortunately, I was not able to spend my entire internship hours with Dr. White because she's a very busy lady. She's a world-renowned virologist and she travels the country. I think I only met with her 4 times because she was constantly in Russia and she was constantly in Libya and New Guinea, bringing back like the knowledge that they have and the tests that they were using with the birth control medicine and the different hormones for fertility drugs and things of that nature. So I had to pursue another internship in a field that was close to virology, and I had 2 choices between infectious diseases, which is a doctor that you see in a hospital who will tell you you have Ebola or tell you you have the flu, versus a pathologist, which is a post-mortem form of virology where they look at cell cultures and they retrieve different things from dead bodies to tell you if that person had Ebola and if they potentially were patient 0, which is how I found my mentor, Dr. Beatrice Lopez. Dr. Lopez is the chief of neuropathology and autopsy at the University of Virginia Hospital, and she is from Brazil, so she gained her education in pathology differently than we do in the U.S. She had to start her college years when we were sophomores, in a sense, and she did an internship and figured out that she wanted to be a pathologist. And she came over to the U.S. through a dual internship program with med school related stuff. And she eventually liked this area and stayed here, and now she's the top of her field. Dr. Lopez works on brains in specific. That's her choice of focus. And her department is huge. There are like 7 different areas of pathology that you can study in, and she chose brains because brains are awesome. And so the first thing that I did when I was with Dr. Lopez was I watched 4 brain autopsies. 2 were on adult males, one who died from complications a week after having a heart attack, and another who died from trauma faced in a motorcycle accident, and then 2 stillborn infant brains. The adult male the first brain that we looked at was the adult male who suffered from the heart attack, and it's similar to the picture on the screen here. You see the different discoloration of the brain. It's supposed to be pink, and his brain was really dark, brownish, dark blackish, which indicated to them that he had suffered from a heart attack because you can see the strains that you put on your body in your brain once you die. And they cut his brain open. Before they cut his brain open, I was allowed to hold the brain, which was really interesting because the brain was soft but squishy at the same time, or really heavy but light. So it was a really weird feeling to hold a brain and disgusting at the same time. And so they had to cut the brain open to do further research, which is what you can see here. We have the slides and the different sizes that they have. So they cut the brain in half using a deli knife, and it like slices like deli meat, which is really weird. And it falls apart off the brain. So they concluded that he had died from complications of the heart attack after they sliced the brain, and they moved over to the motorcycle patient, and his brain was completely falling apart. It had no structure whatsoever, and the atmosphere in the room changed when they brought that brain in. It really smelled like formaldehyde, which smells like mint gum. And the room started swirling for me, but I was still okay at the table then. And then his brain fell apart. And I wasn't exactly okay then, but I lasted through that one. And then they brought out the infant brains, which looks like white mucus, and it fit in the palm of my hand when I held the second one. And it fell apart and like completely disassembled in my hand, and that's when I was passed out. And I sat out the rest of the day when they were doing the autopsy so that they continue. And the next thing that I did with Dr. Lopez was we went upstairs to the surgical area of the pathology unit. So a pathologist still can work on live human remains before they're exactly processed and put into formaldehyde for further processing and soaking. So they were working on 2 things. They were working on a uterus and a liver. A liver is supposed to weigh 150 grams, and this liver weighed 2 kilograms. So it was about this big, and it was lumpy and pussy and really disgusting. And when Dr. Lopez cut it open, the blood and everything spewed everywhere and got all over her. So I had to sit there and wait for her to change. And while she did that, one of her residents was cutting a uterus, and you could see like the different structures as they cut it open. And it was really interesting to see what that looks like, but disgusting at the same time. Basically, my day was very disgusting and gross. So that was basically all that I did for my internships, and I got all my 10 hours doing that amazing pathology work and studying virology, which leads now to my community service. I also did my community service like Hunter at Ronald McDonald House, and this is Miss Coffee. And I did a lot of different things compared to some other people. I cleaned the desktops and I did more computer work because I'm computer savvy. I filed papers. I also laminated books, which is very difficult. I completed their entire library project and I laminated 600 books by hand without a laminating system. So you have to tear off the pieces of laminate, then put it on the book, make sure there's no air bubbles, and then label it and then put it in the system. But it's really nice to see that they have all this cool stuff for the people at the house to come back and look at. So if their child is, say, sick or has to wait for a transplant at surgery, they can check out a book there instead of having to go to a place like our Jefferson Library or come to Fluvanna or somewhere else. Because they house patients from as far as Tokyo to California. So anybody can come here and do anything that's needed for their child, and it helps them because they don't have to pay the expensive hotel prices. Like for instance, one of the couples that was staying there had an infant child who had 6 surgeries since the time they were born, and she'd been there in the UVA hospital for about a half of the year, and they stayed there over 180 days, which is twice the amount of days that they allow you to stay. But at Ronald McDonald House in Charlottesville won't kick anybody out regardless of if they go over their 90-day limit. So their bill was very expensive, and this lady called one day that I was manning the desk, and she completely paid for their entire bill, and they stayed another 20 days more after they stayed, and she paid for that too. So the family didn't have to have a burden at all to think about that, and to see their expressions when they came back and found out that their bill was completely paid for really inspired me to help people in our community by doing different things like my legacy. I worked with Hunter on our legacy is called Stuff with Love. It was a stuffed animal drive that worked in conjunction with Gleaning for the World's Teddy Bear Brigade. This is my heart and joy of the project. And my club, the Future Business Leaders of America, are going to see the project through for the next 5 years. And I'm happy about that. And I can't wait to tell you all about all the cool people we had donate to this drive. We had Gina Keller, our superintendent. We had Mike London and his family. It was really interesting when Mike London donated the teddy bears because I go to the same beautician as his wife and daughters. And I was talking to his wife under the dryer about our community service project and the project as a whole. And she was talking to me about UVA and how great it is and how I should carry that project over to UVA. But I'm not going to UVA, I'm going to Tech. So I had to break that to her. But she still donated, even though Tech football is better. So it worked out. So she donated 75 bears total, and we had a total of 300 bears altogether, which is approximately 3 shipments that can be sent out. The one thing that I liked about Gleaning for the World is they don't just donate to children in the United States who are suffering from a disease. They donate to people who have disasters. Like for instance, our shipment of bears that I checked with this morning will go to Chile since they just had a volcano eruption. So children in Chile who are being evacuated will have stuffed animals to comfort them. I mean, we've all had that stuffed animal that we hung to when we were a child, and it's great to give this opportunity to different people. Furthermore, I have one piece of advice for you all. Think outside the box. I had no interest whatsoever in pursuing a degree in medicine, but now I'm thinking about changing my college choice to the field of medicine instead of engineering, or ways that I can combine them. But without this opportunity, I wouldn't have been able to see that. And you just think outside the box when you're choosing. Try to be different. I mean, everyone here that you've heard so far had a different internship with different opportunities and different things that they were involved with. So it's possibilities are limitless when it comes to this internship process and the community service that you could do to help your community. And that is all I have for you all today. That is the end of my presentation. And are there any questions?
Sasha, I have one. There's been a lot of stuff in the news recently about them manipulating like polio and HIV to fight cancer. Is there any concern that taking viruses that currently we see as somewhat deadly and using them as a cancer fighting agent, that they could then mutate again and we'd have a super polio going around?
There is, and then there isn't. Like, for instance, Dr. White's lab worked with cancer cells in earlier trials when they were trying to find a cure for Ebola, and it sort of mutated the wrong way and it didn't work out. And then on the other end, there were some doctors who were fighting Ebola with the flu because Ebola has the same symptoms as the flu, and sometimes when viruses combine and they mutate with their genome structures, they sometimes their symptoms lessen and the things that infect them changes, so it works out either way.
How did you disconnect in that autopsy? How did you disconnect from patient to body or brain? How were you able to do that at all, or did you have to kind of talk yourself through that?
I didn't have to see a patient like in specific. I just saw a dead brain. But I'm the kid that wouldn't dissect a frog in 10th grade. So seeing that scene and it once it got gory and science fiction-y, that's when I disconnected from it being a patient to this is real life. This is what people go through. This is how people die. It was an overall weird experience.
And Dr. Lopez, were you surprised how much access you had when you were working with Dr. Lopez?
Yes. She told me that I could cut the brain when they were doing it because it's weird. It's different from working with a live patient. Since it's a dead body, you can really do anything as long as it's ethical. And it's a learning experience. So since she's the chief of her department, she would have to approve it. So basically she approved it while I was here. So I was able to do it.
There you go.